Hello to everybody. My name is Nicola Fiore and I am a professor at the University of Chile. This is a brief presentation about the situation of Phytoplasmas infecting grapevine in Chile, including some results of the epidemiological studies that we have carried out thanks to the financial support of the European Union. This is the project named TROPICSAFE. As you probably know, viticulture is one of the most important agricultural activities of Chile, and Chile is the leading wine exporter in America and the fourth world exporter. These are the main symptoms associated to Phytoplasmas in grapevine plants in Chile. They are not different from those that could be observed in other parts of the world. It is not possible, only through the observation of symptoms, to identify the phytoplasmas that are infecting a grapevine plant. Laboratory analysis is necessary. Leaf curl is the typical symptoms, and in white cultivars, the leaves turn yellow, while in red cultivars, they are reddish, including the veins. But the most important problem is the drying up of the bunches in several wine grapes varieties. This starts a few weeks before the physiological maturity of the bunches. In Chile, the grapevine is affected by 6 phytoplasmas that we found in several vineyards. The first two phytoplasma are the 6CSR1B and 1C associated to the Aspergillus group. The second, or the third really, is the 3J phytoplasma associated to the X disease group. Other one is the 6CSR5A phytoplasma associated to the Helm-Jellos group or belonging to the Helm-Jellos group. Other one is the 7A Candidato Phytoplasma fraxini, and the last one is the Candidato Phytoplasma solani, or also named Stolbur or Bois Noir. Between these phytoplasma, the most important one is this, the 16SR3J phytoplasma, because it's the myphytoplasma detected in the last 5 years in the grapevine yellow infecting vineyard in Chile. It is endemic and widely distributed in the American continent. Fortunately, the flavescens doré phytoplasmas and its vector Scaphoideus titanus have not to be found in Chile. As I told you, the 3J phytoplasma is the most widespread in Chile, both in grapevine and other plant species. This is a vineyard planted with Pinot Noir, with the typical symptoms of phytoplasma infection. And then in the same vineyard, we have detected this same phytoplasma in 2 weeds, Polygonum aviculare and Convolvulus arvensis, but also in a leafhopper, Paratanus exitiosus. The same phytoplasma, 3J, was also detected in other weeds, shrubs, and no grapevine woody plants from different botanical family present in vineyard and in their surroundings. Also, as a malva, as you can see, Brassicarapa, Rubus ulmifolius, with the typical witch's broom, and also Rosa with witch's broom too. These plants are a reservoir of the phytoplasma 16 SR3J. To determine if an insect is vector of a phytoplasma, it is necessary to carry out the transmission trials. For this, the insects are placed to feed on phytoplasma-free plants. In this case, we have carried out and concluded the transmission trials and have confirmed that the leafhopper Paratanus exitiosus and Bergallia valdiviana are vector of the 3J phytoplasma. Bergallia valdiviana is another abundant leafhopper present in vineyards, but less than Paratanus exitiosus, of course. These are some symptoms in periwinkle plants. No? The insect have been placed in an entomological cage with periwinkle and the grapevine plants to feed on it. And in these cases, you can see the symptoms associated to the transmission of 3J phytoplasma in periwinkle plants. We have here yellowing, witch's broom, virescence, and phyllody. These insect species, Paratanus exitiosus and Bergallia valdiviana, have been able to transmit the 3J phytoplasma also in grapevine plants. You can see leaf deformation, short internodes, and double node. Symptoms in periwinkle appeared at 3 to 6 months from the start of the transmission trials, while in grapevine plants, this occurred after 1 to 2 years. We found in grapevine, really in vineyard, we found several other insect species of leafhopper that tested positive for 3J phytoplasma. Here we are 5 of them as Bergallia. We have to determine the species, identify the species, but Amplicephalus ornatus, Amplicephalus curtulus, Amplicephalus pallidus, and Exitianus obscurinervis. With this, all of these insects, we are performing the transmission trials to find out which of these species is vector of the phytoplasma 3J. According to the first results obtained, all of these insect species seems to be able to transmit the 3J phytoplasma in both periwinkles and grapevine plants. The insect captured in Chilean vineyards do not represent a direct problem for the crop. The insects generally live associated with spontaneous plant species and only occasionally feed on the grapevine plants, transmitting phytoplasmas. Apparently, the presence of few individuals contributes effectively to the spread of phytoplasmas in the vineyards, even more when infected spontaneous plants are found in the field. Therefore, the indirect damage that this insect causes to the grapevine should not be underestimated. On the other hand, spontaneous infected plants present in or in the vicinity of the vineyard are important reservoir for the 3J phytoplasma. Concluding, for the control of phytoplasma, it's important to perform an appropriate agronomic management of spontaneous plants and leafhoppers associated with the vineyard. And we must not forget the use of phytoplasma-free grapevine plants during the planning of a new vineyard project. Thank you very much and goodbye from Chile.